Hey everyone, welcome back to another video. In today's video, we're going to be continuing the nutrition in mammals with reference to monomers and polymers. It's also useful if you take chemistry. So monomers are small molecules which have their own identity, but polymers are when many monomers combined together. Let's write their definition. Monomers are small molecules that combine to form polymers, and that can't be broken down further. You can't make a smaller, simpler molecule out of a monomer. So they can't be broken down further. When we talk about polymers, it is again a reverse definition where we say these are long chain molecules made by the repetition of monomers. Now, when we say the repetition of monomers, we should understand that it can be in a continuous pattern also. We'll come across this idea. So monomers combine to make polymers. This phenomena is known as condensation, where many molecules combine together. The reverse of condensation is hydrolysis. In hydrolysis, a polymer is broken down into its original monomers. Like imagine these three different molecules which are actually amino acids. Each amino acid is represented by a different color, but you can see they have specific groups on the corner. The COOH atomic combination is known as carboxylic group. You will come across this in the last chapter of chemistry, don't worry about it. And this NH2 part is known as the amine group. Every amino acid has this. And when they combine in a specific manner to make a protein. You don't have to learn this, but you should understand how monomers are combining. You can see that they have a linkage between them, which is known as the peptide linkage. It never comes in O-level bio exam, but it would be really good for your understanding of what's actually happening on the molecular level. So amino acids combine to make proteins by the process of condensation, and they make peptide linkages. Here peptide linkages are formed, they are made. If somehow I want to break the protein back into the amino acids, that would be called as hydrolysis, where I would have been breaking these peptide linkages. So peptide linkage is broken in hydrolysis and they are made in condensation. Another monomer you can see over here is a monosaccharide. A monosaccharide means single sugar molecule. These hexagonal molecules are actually glucose. When they combine together, they make a polysaccharide. You can see the word also. Mono means one sugar, polysaccharide means many sugars. So many monosaccharides combine to make a polysaccharide, for example, glycogen or starch or even cellulose. These sugar molecules, when they combine together, that's process of condensation. We know that in condensation, monomers combine together. Polymer breaking down would have been hydrolysis. Here, the linkage is known as glycosidic linkage. Again, you don't have to know this for O-level bio, but it would be good for your basics. So glycosidic linkage is made in monosaccharides to make a polysaccharide. Like in proteins, it used to be called as peptide linkage. A third example of condensation and hydrolysis happens when you have three different molecules known as fatty acids and the fourth molecule is known as glycerol. When one glycerol combines with three long chain acids, it is known as a lipid, or you can say fat or oil. The actual word, biological word, is lipid. Three different fatty acids combine with one glycerol in condensation to make a fatty or to make a lipid, and then lipid can break down through hydrolysis to give you back those four molecules. In lipids, the main linkage is known as the ester linkage. Now let's deconstruct and go into a deeper level of our understanding, starting with carbohydrates. The word carbohydrates mean they have carbon, hydrogen and oxygen. They are compounds made up of monosaccharides. Monosaccharide is the monomer of all carbohydrates. So they are compounds made up of monosaccharides which contain C ratio H ratio O in 1:2:1 ratio. So that is fixed for monosaccharides, remember that. If you were in A level, the definition that you would be expected to know is that they are polyhydric aldehydes or ketones. They are polyhydric aldehydes or ketones. That's the correct definition. I always teach it to O-level bio students also, but you can choose to write the blue one also because at O-levels that's okay. Let's understand monosaccharides more with reference to disaccharide and also in comparison with a polysaccharide. You can see the word saccharide is constant, which means sugar. Mono, di, and poly are the changes here. Monosaccharides are single monomers. Disaccharides have two monomers joined together through the glycosidic bond. Polysaccharides have hundreds of monomers joined together by many glycosidic bonds. Monosaccharides are mainly crystalline, and even disaccharides are crystalline in structure. Polysaccharides are amorphous, which means they look like white powder. Monosaccharides have mainly sweet taste. Disaccharides are not always sweet. Few of them are. Polysaccharides are tasteless. So if you were to taste starch, it would have no taste at all. Monosaccharides are mainly soluble in water. So when you make glucose water, it is basically a monosaccharide solution. Disaccharides are also soluble. Polysaccharides are insoluble in water. When we try to understand them more, we should know that there are categories of even monosaccharides on the basis of how many carbon atoms are here. If there is a 3-carbon sugar, for example, with the formula C3H6O3, it would be called as triose. A 4-carbon monosaccharide, C4H8O4, would be known as tetrose. C5H10O5 sugar would be known as pentose. Pent means 5. And a C6H12O6 sugar, which would be known as hexose, because hex means 6. Pentose sugar is very important in our body, which makes ribose for our DNA molecule. Hexose sugars are glucose, which we need for energy, fructose, which is normally present in fruits, and galactose, which is present in milk. Do we have disaccharides? So there are few common disaccharides also. Disaccharides are made when two monosaccharides combine, so they have the formula C12H22O11. It is made by the combination of two monosaccharides. This particularly is made by the combination of two hexoses because it is C12, so 2 times C6. A very good example is sucrose, which is present in the table sugar that we use. It's a combination of glucose and fructose. We got lactose, which is present actually in milk. It is a combination of glucose and galactose monomers. Another very common example of disaccharide is maltose, which is made by the combination of two glucoses. There are other disaccharides also, but we don't need to know about them. A polysaccharide is a combination of hundreds of monosaccharides. The most common example we have for a polysaccharide is starch. In the previous chapter, we have talked about starch a lot. It is present in plant roots, especially as a storage of food. So thousands of glucoses are combined to make starch, and then we store it. Another example is glycogen. Glycogen looks very similar to starch, but animals use glycogen and they store glycogen inside their liver. So present in animals, example in liver. We store glycogen in our liver as storage. The third example we have is cellulose. We know about it. We know that it's present in plant cell wall. It is there for strength. So cellulose is not used for storage, it's for strength. Now we are at it. We should talk about starch and glycogen a little more. These chains you see are these long chains are actually thousands of glucoses combined together. These chains are known as amylose. Amylose means unbranched chain of many glucoses by glycosidic bonds. But then you can see also branching happening, right? This branching part is known as the amylopectin. So if it's a straight chain of thousands of glucoses, it's amylose, and then the branching is known as amylopectin. Starch and glycogen both have branches, but starch has fewer branches, glycogen has more branches. Starch has lesser density. You can see that starch molecule is very open. Glycogen has more density because it's very congested. In plants, you can see starch, especially in their roots. There's a cell known as parenchyma in their cortex. So, potato, for example, has a lot of starch in it. Beetroot, for example, has a lot of starch in it, stored over there. Where do we find glycogen? So glycogen is in animals. Example, in our liver. So when we eat food, when we have a lot of glucose taken inside the body, our body combines so many glucoses and makes glycogen in our liver and muscles. Cellulose does not look exactly similar to starch and glycogen, only that it has also glucose. It's present in plant cell wall. So there you can find cellulose fibers giving strength to the plant cell wall. So it provides strength and doesn't allow them to burst. If you put a plant cell in water, it's not going to burst because of the cell wall. Let's talk about amino acids and proteins in a similar way so that we know what's happening. An amino acid is a molecule which has one carbon bonded to COOH, as you can see on the right side, and NH2 as you can see on the left side. The third bond is a simple hydrogen and the fourth bond is variable part. NH2 is known as the amine group and COOH is known as the carboxylic acid group. Every amino acid has it. Every amino acid has a variable region known as the functional group, and that is why amino acids are different. Each amino acid has a different R group. There are 20 natural amino acids present in living organism, each amino acid with a different R group. So we have many synthetic amino acids also where we make the R group ourselves in the laboratory, but those aren't natural. Moving on. When we talk about amino acids, they combine to make longer chains. Through condensation, we know that. And that when this sequential condensation happens, we get a sequential chain known as the polypeptide chain. Poly means many, peptide means the bond. So many amino acids combine through this condensation to make a polypeptide. And when you have so much longer polypeptides combining together, you get a very complicated protein. A protein is made by hundreds of thousands of amino acids. There are times when there are 5, 6, 7 polypeptide chains together. For example, hemoglobin has 4 polypeptide chains. It's really huge. Moving on to lipids. Lipids are made when there are 3 fatty acid molecules. They combine with a glycerol molecule to make a compound called as the triglyceride. The word tri means 3, glyceride means fatty acids, or you can say esters. So there are 3 fatty acids and 3 ester linkages. It's very important for us to know that in a triglyceride, 3 fatty acids combine with 1 glycerol. The fatty acids can be of 2 different categories. The first is saturated lipids or saturated fatty acids. The other is unsaturated lipids or unsaturated fatty acids. If your fatty acid, Fa here means fatty acid, if your fatty acid has no carbon-carbon double bond anywhere, it is called saturated. If fatty acid has 1 or more carbon-carbon double bonds, it's going to be called as unsaturated. The saturated ones have higher melting points and boiling points. The unsaturated ones have lower melting point and lower boiling point. The saturated ones are mostly solids at room temperature. For example, when you look at ghee, it's a kind of a saturated lipid. But the unsaturated ones are mostly liquid at room temperature. For example, all the oils, they are mainly unsaturated. The saturated ones are mostly present in animal sources. For example, animal fat present in meat, beef, for example, animal wax, that's saturated. The unsaturated one is present in plant sources. For example, soybean oil, olive oil, sunflower oil, these are unsaturated lipids. I hope these ideas are clear for you guys. Stay tuned. Thanks.